Welcome to What's New in MPMI, the virtual seminar series of the MPMI Journal. Glad to have you with us. We've had a little break and we're back in gear. Very excited about today's seminar. While people are joining, just to remind you, our questions will show up in the Q&A. You can click on the Q&A at the bottom. You can write a question at any time. But while people are joining, why don't you type in the chat where you're from? It's always exciting to see our international community and where everybody's joining from. So I'd love to see that. So please go ahead and type that in. And once people are on, I see people are still logging on. We'll wait about a minute and then we'll get started. So I can tell you good evening from where I am, but I know good morning to our speaker, Yusuke Saijo, and co-host, Kenichi Tsuda. Where are all of you from? I see Lynn Epstein is from Berkeley, California. We're getting these in the Q&A, but you should feel free to also type in the chat where you're from. Somya Pusapati from San Diego, California, welcome. This is one of our Pacific focused seminars. So the timing is adjusted to be great for California, for Japan, for China, for Australia. So I'm hoping we're pulling people from all these different places. Okay, well, welcome to What's New in MPMI. As I said, I'm Jean Harris. I'm editor-in-chief of the journal. With me as co-host today is Kenichi Tsuda, who is guest editor of this focus issue, which we are highlighting today, the focus issue on top 10 question number 2. How does the abiotic environment influence plant microbe interactions? And the answer is very complicated, lots of interactions, lots of different ways. And today's speaker is Yusuke Saijo, who is from the Nara Institute of Science and Technology in Japan, and who was the senior author of our editor's pick, one of the most exciting papers that came out in our recent focus issue. And I think that we will post in the chat where the focus issue, the link to the focus issue, because I hope that you will explore not only this paper, but also many of the others. So the way this will work, obviously we're going to have our seminar by Yusuke Saijo, and at the end, we will read Kenichi Tsuda, my co-host, and I will be reading your questions that you type into the Q&A, and we'll answer them. And we really love this part where we have the interaction from the community. We have an international community, and I can see already that we have people from around the world, which makes this really worthwhile. I also want to highlight before we go on to today's talk that our next seminar will be November 15th, and it will be our graduate student award winner, Kyung-Young Seong, who worked with Ksenia Krasilova to write the graduate student award winning paper and on using machine learning to detect and predict different effectors. So that was a very cool COVID project that he did, and I'm looking forward to that one as well. Okay. So just a last reminder, type your questions into the Q&A. You may do it at any time. About 24 hours after this seminar is done, we will try to have that posted online. And we are also looking for volunteers to help us edit the transcript. And this helps us to increase accessibility to people who have any kind of hearing impairment and to people for whom English is not their first language. If you would like to do that, please put your name in the chat to Karen or to me, and we will contact you. We really appreciate volunteers who have helped us to do this. It's a real plus to help to make this more accessible to our entire audience. Okay, so you can see in the chat where to find the focus issue, all the other papers, including this one, and the recording of the webinar. And I am going to turn it over now to Dr. Saijo, Yusuke Saijo, who will be talking to us today about a really fascinating paper, caught my eye as soon as it was submitted, recognition of microbe and damage-associated molecular patterns by LRR pattern recognition receptor kinases and how they confer salt tolerance in plants. Fascinating story, wonderful crosstalk, really illustrates how complicated these interactions can be in the real world. I'll turn it over to you. Thank you, Yusuke.
Thank you very much for nice introduction and giving us an opportunity to share our studies and paper. So good morning and good evening, everyone. So I hope, today we can share a great time. So I'm Yusuke Saijo from Nara. So you can see the photo. So where our campus is located. So I like to first introduce some backgrounds of plant microbe interactions. I'm aware that so most of people know this, but let's make sure the background. So first, plants in nature live in close association with many diverse microbes which inhabit on the surface or inside the plant tissues, and which really greatly influence the plant survival and also health, adapting to different environments, and of which some become pathogens, they're causing some harm and diseases on the plants. And then, so how plants sense and manage these biotic interaction with these microbes is dependent, largely dependent on the plant immune system, which consists of two major classes of immune receptors. So the one is the pattern recognition, which act on the cell surface, detecting microbes or molecular structures typical of microbes or derived from damaged host cells. So these patterns are recognized by so-called PRRs, pattern recognition receptors, and which leads to so-called pattern-triggered immunity. So this is basically effective against most of infectious microbes. So to counteract this layer of plant immunity, so adapted microbes, so including pathogens and also commensals and also mutualists, seems to use effectors to perturb this immune layer and also manipulate the host cellular process and to promote infection. In turn, plants have also evolved the second class of immune receptors, so predominantly provided by intracellular NLRs. So this provides a kind of backup immune system to detect pathogen effectors and to trigger more strong and robust immune responses called ETI. So the recent studies have shown kind of functional linkage and interdependence between these PTI and the ETI. But the molecular details are still, require further investigation. So I would like to extend this kind of, history of plant immunity studies to this kind of important, the question of plant microbe environment interactions. So although, our kind of molecular microbiology or molecular knowledge accumulates in plant microbe interactions, but how they are influenced by different environmental factors and fluctuating environments is still poorly understood. And in this context, I hope our study, today I talk, will give some insight into your this kind of aspect of plant biology and also stimulate your further studies. Okay. So what happens when pattern recognition receptors are activated when they recognize cognate ligands? So I'll take example from kind of the best characterized pattern recognition receptors which detect the microbe signals like bacterial flagellin or EF2U. So the cognate receptors FLS2 and EFR respectively, leucine-rich repeat receptor kinases, and then they instantly form receptor complexes with co-receptor background following the cognate ligand recognition. So leading to the formation of the receptor complexes involving RLCKs, receptor-like cytoplasmic kinases, like BIC1 or PBL1. Later I use. And then so following this ligand perception, there is a common set of immune responses activated. So kind of you see a basic principle. So following the pattern receptor recognition, so these kind of common set of defense responses are activated, and they are collectively contribute to enhancing immune state of plant immunity. So these pattern receptors can be considered as a kind of inputs of different stimuli, particularly immunogenic ligands, and to the activation of a cellular state of immunity. And then now you see the expansion, large expansion of receptor kinases and receptor-like proteins, and most of which are likely receptors, immune receptors, and then likely acting as pattern recognition receptors. And then this really expansion suggests that plants really rely on these receptors to sense and manage biotic interaction, and also likely kind of abiotic stress responses, and then to better adapt to the environment. So in addition to, so among these receptors, so in addition to the receptors detecting microbial signals, there are some also receptors which detect the endogenous immunogenic signals produced by the plant. And here, so I just show one example. So in which, MAMP receptor activation leading to the activation immunity, which involved the kind of peptide precursors, so that released the following kind of cellular membrane damage to the external space, and thereby being detected by another receptor called PEP receptors. And then this recognition of the peptides leading to the further activation of immune responses. So in this way, plants may sense the kind of damage which cannot be recognized only by monitoring the microbial signals. So it can be said that plants can sense the kind of the presence of pathogens or really severe threat, and requiring their strong activation of immunity when detecting these microbes and damages, and then which is associated with pathogen infection. So, but in the course of study, so we initially pursued how this system really acts to activate the pathogen resistance. And in the course of studies, so we just put a particular focus on these peptides, which are now well characterized, and they are called propeptides. So which have the elicit active epitopes in the C terminal. And of these 8 members so here I show only 6, but there are 8 members propeps in Arabidopsis, and of which and 3 are massively induced during pathogen challenge. And then these peptides are like flagellin peptide or alpha-18 peptides. So their application also activate immune responses in the plant through the cognate leucine-rich repeat receptor kinase receptors, PEPR1, PEPR2. But so these peptides don't have any terminal signal peptide, so which is required for conventional secretion. So that's why they are thought as a kind of damage signal, which for the first time released when cell membranes are disrupted. So then their perception can be interpreted as a kind of the detection of cell damage. So following this, so recent studies have shown the metacaspases involved in the cleavage of this peptide. And then in parallel with these studies, we also show that some members are released. So even without to the extracellular space, where they are recognized by the cognate PEP receptors. So initially, when we started this study, so we just wanted to know how the plants recognize pathogen damage and pathogen challenge. And in this context, we examined so how and whether and how so this ProPEP is released to the extracellular space in response to pathogen challenge. And then to this end, so we subjected the transgenic plants expressing ProPEP3 under their own DNA regulatory sequences, and then fused to Venus, GFP variant, and then to monitor their production and release by subjecting the seedlings to kind of different, Schulman syringe bacteria, and see what is expressed inside plant tissues and also released to the cellular kind of space, which is in the media in this experiment setting. And then, so in these settings, we show that, so the implant, so this precursor is indeed induced in response to virulent bacteria and also ETI triggering virulent bacteria, okay? And then, interestingly, so they are released to the extra space, specifically following this virulent bacteria and also ETI triggering bacteria. And then further evidence also suggests that so their peptide release is associated with not only with the pathogen resistance activation, but also with host cell death and damage. So this is consistent with the view that they provide so damage signals. But now then, when we saw this data, and then also were aware that kind of cell damage is quite common, not only to the biotic stress, but also to abiotic stress such as salt. And then we looked at the transcriptome responses to these PEPs or allohating, so these dumps, and which typically induce common set of defense-related transcriptome. So at that time, so my postdoc, so we actually, so today's topic is what's new, but we, first, obtained these data already 10 years ago. And then at that time, so we worked with postdoc Koji Yamada, who has later discovered the sugar transporter as an important, phosphorylation target of pattern receptor complex. But anyway, at that time, so because he used to work on, the salt stress responses, and he immediately kind of noticed that the high overlap between these MAMP and DAMP induced transcriptome responses with salt induced transcriptome. So as you can see here, that most of those PEP or ELF induced genes are highly induced also in response to salt stress. And then at that time, we are very focused on these PEP receptors and to examine a role for damage sensing in the modulation plant immunity. So we are particularly interested in this kind of damage sensing. And then we noticed that, okay, these pro-PEP families and also PEP receptor genes are also highly induced in response to salt or osmotic stress. So then, which kind of, inspired us to test possible dependence of these peptide receptor system in plant salt tolerance. So to test this, we just undertook very simple approach, and by pre-treating seedlings with PEP and peptide, and then some days after, transfer the seedling to the media containing high concentration salt. So usually, so Arabidopsis seedlings die when exposed to like more than 100 millimolar, 175, but this could depend on some other conditions. But anyway, plants die if you don't do anything, But then, but if you pre-treat seedlings with this peptide, and you see a dramatic increase in the survival rate, and then we got, okay, so it's likely that, okay, so pre-activation of this pattern receptor signaling can enhance salt tolerance. And this kind of initial observation has been followed by more detailed studies in Germany. At that time, I was in Max Planck Institute in Germany. And then after I came to Nara Institute in Japan, and in both places on the planet, not only in Germany, but also in Japan, we observe the same response you see here. So when seedlings are treated with PEP2 and then wild-type plants, acquire salt tolerance in a manner dependent on the cognate pattern receptor PEP2. And this is also the case when treated with PEP1. And you see this kind of time course. And indeed, so the PEP1 treated wild-type plants retain high survival rate. In contrast, so the mock controls and also receptor mutants did not, acquire the salt tolerance. So this survival rate is, supported. The increased survival rate is supported by, another set of data that, for example, seedling retain growth under salt stress condition to some extent very well. And also, if you look at chlorophyll contents, which is well retained in, PEP-treated white plants. So this strengthens the idea that so, pattern receptor elicitation result in the increased salt tolerance. So we call this phenomenon as a pattern triggered salt tolerance. And then today I just call PTSD. Okay. And then so this concept, this view is also extended to bacterial mumps like FLAG22 or F18. So this FLAG22 and F18 treatment, pre-treatment also increased the salt tolerance of seedlings in a manner dependent on cognate receptors, FS2 and EFR. And we also examine the dose dependence of this FLAG22 induced salt tolerance and found that so this is quite similar to what we are familiar with in their defense activation. So we conventionally use like from 0.1 to like 0.5, 1 micromolar of FLAG22 to make sure strong activation of defense responses. But usually we observe some extent of activation, let's say like this, molar range. Okay, so this is quite consistent with also defense activation. And then, but you may be kind of careful, and you might say that one, the pattern receptor, triggered outputs includes, growth inhibition, which is prominent when young seedlings are exposed to these, mumps or dumps under sterile conditions. So you may say that so the growth inhibition then reduces kind of metabolic activity, and which may in turn reduce the unwanted uptake of salt from the medium. Okay, but we tested this by testing kind of different settings in which, for example, PEP3 and PEP4 induce defense responses and also salt tolerance, like here, without compromising root growth. Okay, and also root growth inhibition in response to peptide is mainly dependent on PEP receptor 2. And then so these single mutants will retain growth, but this can still acquire significant salt tolerance. So indicating that so the PEP receptor both contribute to salt tolerance in response to PEP peptide, and also that growth inhibition is not required for salt tolerance, PTSD. And then we extended our studies to test the genetic requirements for PTSD. And we initially tested these kind of well-known components involved in the receptor complexes. So including co-receptor background and also the immediate downstream regulator receptor like cytoplasmic kinases, BIC1 and PBL1. And then when you see the result, so the null mutants of background, so as previously shown, in our study, that so this peptide signaling relatively retain the kind of resilience to the disruption background by using different background-related co-receptor kinases. So this is not really unexpected, but this is kind of expected, okay? And then also, but importantly, so in background-5, so hypoactive, background allele, so which specifically disrupt the pattern co-receptor function. And then in this case, you see a dramatic decrease in PTSD. And this also is observed in also BIC1, PBL1, and also these double mutants. And then, so making sure that, okay, these authentic receptor complexes components are required for PTSD. The furthermore, kind of one kind of hallmark defense outputs of pattern receptor signaling involves the RBHD induced loss burst. And then the data suggests that so this RBHD also contributes to PTSD. But in contrast, so PMR4 dependent callus division seems not required for PTSD. Yes, so but here I also like to stress that when you see the mock samples, mock values. So for example, if you look at the core receptor mutants and these RSAK mutants, so without PEP application, these seedlings are already more susceptible or sensitive to stress. So suggesting that so the somehow endogenous ligand or endogenous signal and receptor system seems to be involved in salt tolerance as well. And then we tested and show that so this is also the case for fructane-2 induced PTSD. So by testing a series of mutants and in fructane-2 induced PTSD. Interestingly, so I also noted that so the BAC1-4, so this BAC1 mutants are well known as fructane-2 insensitive, but in this case somehow unexpectedly and honestly it's puzzling for us, but somehow this PTSD in response to fructane-2 it's retained in BAC1-4 mutant. I don't know why, but this also reflects the kind of robustness of pattern receptor triggered salt tolerance. So this robustness is also further supported when we tested so different hormone related mutants. So first we took this DD2 IN2 PAD4 CYT2 quadruple mutants generated by Kentsuda. So you can see here. So in this mutant, so essentially all defense related JA, SLN, and SA defenses simultaneously disrupted. But even in this mutant setting, so you see a high activation PTSD following PEP pretreatment. And moreover, so when it comes to salt osmotic stress, and everyone see important role for ABA. But in this case, when we tested, so ABA biosynthesis mutants and also ABA regulating important transcription factor mutants, we see a wild type like PTSD. So indicating that so this PTSD is quite robust also against the hormone perturbations. So I switched the read gear and to see how this PTSD happens with a initial focus on again our familiar this ProPEP or PEPR system. And first of all, so following the data that so even without exogenous application of peptides, the BAC1-5 BKK1 or BCON-PBL mutants are sensitive, lowered in salt tolerance, and then giving an idea that okay, indeed endogenous dump system should work. It's not really an artifact you can see only when you supply high dose of MAMP or dump peptides, and then we just like to obtain evidence that endogenous this system is really contributes to the salt tolerance. To test this, we first examined whether the ProPEP3 is also produced and released under salt stress. And you can see here, so under microscopy, so imaging studies, you can see the high induction of ProPEP3 venous in response to salt stress, in this case roots. And you can see here, so the roots are well damaged. You see the compared to the mock control, so without salt, with salt, and you see really damaged roots here. And consistent with the induction of such damage, we also see in the immunoblot analysis for the growth media surrounding seedlings. So we took this as a kind of extracellular fraction of plants, and then we see in that fraction the accumulation of ProPEP derived peptides. So in response salt. So this indicates that indeed endogenous this ProPEP3 peptide pathway is involved under salt stress. And then we further aim to obtain some genetic evidence for the paper requirements in salt tolerance. And then, honestly, we first simply tested the same setting, but in that setting, we didn't really see the significant difference between wild-type plants and the pepper receptor mutants, which again kind of reflect the robustness of this, kind of PTSD per se. But to test to obtain the evidence for contribution of peppers, so we took kind of different settings. So in which the first, so we transferred young seedlings to mild salt stress and give 1 week for plants to adjust and also adapt and climb kind of this stress condition. And after that, so wild type plants can acquire some the priming effect. But this depends on the accession. So typically in Colombia, so we conventionally use this accession is poor at acquiring this tolerance. But in this setting, so by overexpressing the pepper receptors, we see even in the Colombia background, so in this setting, so this overexpressor can enhance the acquired osmotic stress tolerance. So providing evidence that endogenous peppers also contribute to salt tolerance and osmotic tolerance. And then we really looked at the transcriptome level. So what really happens during PTSD. So in this conventional PTSD assay setting, so we subjected the seedlings. So after PEP1 pretreatment, and when they are subject to salt test, and then to this RNA-seq analysis. So the result first suggested here, so we compare the number of differentially expressed genes, DGs, and then if you really compare the numbers, up or down regulated under salt stress, following like 3 or 24 hours, and then the number, more or less not really reduced. So making sure that this still, in this time window, the salt stress responses still passes in these seedlings. But if you look at the impact of PEP or pepper activation, pre-activation on salt responsive transcriptome, so number of DGs that are dependent on PEP application and also pepper changed during the time window. So you can see the kind of higher impact at the rather early time points compared to 3 hours compared to 24 hours. So we infer from these data that this PTSD effects conferred by pattern receptor or PEP receptor is prominent at an early phase of salt stress exposure. Okay, and then we also notice the kind of the commonality and differences between pattern responsive genes and also salt responsive genes. So by looking at these numbers, so you can see high portion of genes, let's say like 599 genes, commonly induced by peptide alpha-18 or and also salt. But here you can see the really significant number of genes are specifically induced in response to salt or in response to these patterns. So indicating that there is an overlap between pattern triggered and the salt triggered transcriptome, but still separate genes are specifically induced in different stimuli. And furthermore, it's very interesting to us to see that like some number of genes are activated specifically following the PEP pretreatment. I mean, in other words, so without PEP pretreatment, these genes are not responsive to salt stress or even to these pattern dumps, but only in this PTSD setting, so they acquire responsiveness to salt stress. So overall, so these data suggest that this PEP pretreatment has a role in rapid mobilization of salt responsive transcriptome and also its expansion. So which we believe is kind of positive, that contribute to the enhancing salt tolerance. Okay, and then we clustered these genes and they put a particular interest in the cluster number 5. So the, here, so basically, cluster 5, induced in response to salt stress, but their salt induction is accelerated, or this is can rapidly induced only when plants are pre-treated with pepper. So indicating that this represents a class of genes which are rapidly activated after PEP pre-treatment. And then when we look at the cis element, what kind of cis elements are overrepresented in the 1 kilobase sequences upstream of transcription starting sites for these genes, and then the database search found that the high enrichment, particularly of these WAKI class of transcription factors, and then we put the particular interest in this WAKI 40 and WAKI 18. So in the next slides, so this is a kind of enlarged, the magnified picture of cluster 5, and you can note that kind of difference-related genes, which we are familiar and we conventionally use as kind of difference markers, are included in this cluster 5. So somehow cluster 5 genes are related to defense, but their rapidly activation in response to salt is associated with PTSD. And then now, kind of cis elements enriched in their promoter sequences, kind of binding sequence known for WAKI 18 and 40. That's why, so we tested the possible role or environment during PTSD. So first, Arabidopsis, has more than 70 members of WAKI transcription factors, quite a gene family, so involved in different aspects of immunity regulation and also different processes. And then in which, so WAKI 18, 40, and 60 represent a kind of specific subgrade. And then interestingly, they are induced in response to MAMP. So during PTI, so their activation is enhanced, but their negative regulation of, for example, FLAG10 induced defense gene activation has been already described. So then at that time, so we kind of thought that, okay, this kind of negative PTI regulator may have a role in this kind of PTSD. And consistent with this idea that WAKI 40 and WAKI 18, so both protein accumulation, it's enhanced and also more prolonged following PEP pre-treatment. So suggesting that so their enhanced accumulation and prolonged accumulation is associated with PTSD. And then in that accepted paper, we kind of reserve the kind of testing the biological significance of these WAKI 18 and 40, but in a kind of pretty submitted version, we reported their kind of possible role in PTSD and positive role in PTSD. But later we found that so the somehow seed quality used influences the results of our assays. And then we found that so these mutant seeds easily get poor, at least during kind of storage condition in Japan. So somehow we had to reserve this conclusion. But if you are interested, maybe it's worth pursuing their role in PTSD. Okay. So then following the having shown that indeed so pattern receptor activation result in so enhanced salt tolerance as well, not only immune activation. We then thought that okay, if MAMP perception leads to salt tolerance, what happens if plants face to microbes? And to test this idea, so we pre-inoculated seedlings with these bacteria in a kind of spray, inoculation condition. I mean, cell, plant surface, surface inoculated, with these bacteria. So including this herpes mutant, which doesn't have effectors. So they are considered as a non-pathogenic, so less virulent, and then a trigger for PTI. And in addition, we took two virulent bacterial strains, so which activate the cognate NLRs to trigger ETI. So both bacteria cannot, grow well and propagate in the plants. So that's why, so the at least under mock condition, the seedlings exposed to these bacteria grow well, no problem, no difference. But when they are, exposed to salt stress after, bacterial inoculation, then you see that only, increased salt tolerance is observed when pre-inoculated with PTI triggering bacteria, but not ETI triggering bacteria. So indicating that so PTI activation is good at enhancing salt tolerance, but not ETI. So ETI may be too much for defense activation, but not good for salt tolerance. And then we extended this idea and also to test the genetic requirements for bacterium induced salt tolerance. And then consistent with the effectiveness of PTI triggering bacteria in enhancing salt tolerance. So we observed that so the loss of PTSD in this setting, so in the flagellin and the AFTU receptor mutants, as well as in their core receptor mutants. So in this case, bacterium-induced salt tolerance is also impaired. And furthermore, so we obtained evidence using another non-pathogenic bacteria, Schulman's fluorescence, and then this bacterium also induced the PTSD in a manner dependent of cognate, pattern co-receptors of BAC1, okay? And then importantly, we further showed that so even heat-killed bacteria is sufficient to induce PTSD through this BAC co-receptor. So providing, I think, solid evidence that the perception these non-pathogenic bacteria likely through pattern receptors is required and sufficient to trigger salt tolerance when plants are exposed to these bacteria. Okay, so in the end, I would like to summarize our findings here. So first, so we show that recognition of microbial and damage signal induces salt tolerance through pattern recognition receptor complexes, which are required for PTSD. And then in this process, so recognition leads to rapid activation and expansion of salt responsive transcriptome, which we believe is an important factor for enhancing salt tolerance. And in that process, WARKI 18 and WARKI 40 in particular, so their accumulation is enhanced. And in the end, finally, so we also show that so this phenomenon is also true for true when plants faced to non-pathogenic microbes. So given the fact that so plants live in association and host diverse microbes inside their tissues, and then their kind of behavior can be altered so under fluctuating environments. So it's quite conceivable that plants may sense the like salt stress in part through the monitoring the changes in these endophytic microbes. Okay, and then so the paper ends this part, but I like to give some more slides to stimulate discussions after this. Okay, so please stay with me. First, so when we talk this story, then some of you may be wondering, oh, this may be similar to the so-called general stress response, which has been kind of elegantly revisited and shown in these two papers published in Nature Plants last year. So one setting used like 24 genes, setting 39 leaf inhabiting commensal bacteria, and they found like a quite small number of genes are commonly induced when plants are exposed to these commensal non-self microbes. So they're called as kind of general non-self response. And moreover, so the another story, the another paper has nicely shown that so the genes rapidly activate. So in response to, for example, 7 months of dumps are highly similar, shared by the genes induced in different, biotic and abiotic stimuli. So this really points to the existence of some kind of core, kind of stress responsive genes, which establish general stress response. And I feel that conceptual similarity between these phenomena and our findings. But one important thing I like to note is that Although we didn't really examine it in detail, but I expect that some time may be needed for plants to enhance or establish like salt tolerant states following pattern receptor elicitation. So there is really a common, general stress response activated immediately after pattern receptor activation, so which is called general stress response, which is prominent like within 5 minutes or 10 minutes following, the mump or dump perception. But I'm not sure if this is really sufficient for plants to enhance effectively salt tolerance. So this issue, I think, still remains open, but I feel quite high relatedness between our findings of these studies. Okay, and then, so this implies that somehow specific the close relationship between pattern, receptor triggered changes in transcriptome defense responses and particularly salt or osmotic storage tolerance. So in light of that, I like to introduce, so of course, exciting, the previous studies, showing first that the pathogen bacteria, so in this case, they encounter high osmotic stress when plants activate ETI or HRCL, this in particular, but anyway, if plant immunity is enhanced, then kind of osmotic stress imposed on the pathogen inside the leaves is higher, okay? So this also predicts that kind of, microscopic environment, then plants also need to elevate, such high, osmotic stress and so on. So, and then in relating to this, so another also recent, studies have nicely shown that so acquiring, water, the water soaking inside the leaf of plant is an important strategy for, pathogenic bacteria to grow and then propagate and cause disease. And in this context, really water and osmotic, stress or regulation is really key in plant microbe interaction. And then although this study, we show the effects of pattern receptor signaling to enhance salt tolerance at the full plant level, but we should also look at what happens at more microscopically when, plant cells meet, pathogens. And there seems to be some, I think, relationship we should explore in the future. And then in this context, I also like to introduce some studies from our group, then which has shown, so one key output of pattern receptor signaling is to activate and involve sugar transporters and to import sugar from the extracellular space to inside the cells. And this, of course, is described in that paper to play a role in deploying, nutrient, sugar nutrient from pathogenic bacteria. But this also, of course, greatly influences the osmotic, environment in the extracellular space, and which may be also related to the kind of salt osmotic storage tolerance activated following pattern receptor activation. And furthermore, so the extracellular milieu are quite busy. So the recent studies have really expanded the family of so-called, phytocytokine, but anyway, endogenous, immunogenic peptides. And then, so they really act, quite busily, in extracellular spaces. Whether they also play a role in abatic stress responses is also an interesting topic. I mean, some of which has been already shown, but this is also an interesting topic for further research. Okay, finally, I like to acknowledge the people contributing to our kind of old, and I don't know if it's called what's new, but it's for us, it's quite old studies. But first of all, so the Koji Yamada, who has initially discovered this phenomenon already 10 years ago. At that time, we were in Germany. And after that, so we came to Japan, and then to complete these studies, mainly, by the first authors, so Eliza and also Yuri. And then finally, thank you very much for attention. I'm happy for discussion.
Thank you, Yusuke, for an excellent seminar. That was really fascinating. Very excited to hear about all of this. I I'm going to encourage everyone to type your questions into the Q&A, and we will take turns reading your questions. So really interesting to see how you put the whole thing together in the first place. It's something that's very interesting to me because, of course, we've done work on rhizobium, so beneficial bacteria, and seeing a very similar thing, the way its interaction triggers a separate set of separate response in response to salt. So really curious to see how this works with microbiomes, and fascinating to see the things that you've uncovered with that, including those heat-killed Pseudomonas fluorescens. Really, really great. Okay. Time for everyone's questions. What do you have? Type it in. Kenichi, while we are waiting, do you have, was there anything that came up on your end? I know this is very relevant to things that you're thinking about.
So, yeah, while we are waiting for questions from the audience, I can ask some questions. So there has been also cases that like salt stress, abiotic stress, such as salt and plant immunity sort of inhibit each other. But your study showed that plant immunity, especially pattern triggered immunity, enhance salt tolerance. So do you have some idea when this sort of a cooperation happens, when sort of an inhibition happens?
Well, thank you very much for good question. So I missed well explaining that aspect. So indeed, so in depending on the context. So sometimes abiotic stress negatively influences biotic stress resistance or tolerance, and in my view, first this is really depending on the context, and then I have to say a kind of key principle here is that so plants have the ability to fine-tune their responses, and also which to be prioritized, so according to the different factors. So I also noticed in this study that we also found that, so for example, chitin kitchen, the well-known fungal mumps, was previously described in another group to enhance salt tolerance, but under our conditions, we did not really show significant effects. So that's why one reviewer stopped us to use the title of our paper using just pattern recognition receptors, but to specify Leucine-Rich repeat class pattern receptors. But anyhow, I believe the data of chitin-induced salt tolerance should be true, but this somehow our lab condition, we miss some factors which are not recognized by, I'm afraid, anyone that critical for effect. And furthermore, I also like to add to say that we are aware the kind of phasing in immunity regulation defense response. So starting from kind of initial wave of activation, which is nicely described in Dipper's group, that this kind of general stress response are quite rapid, activation of general stress response, which is followed by more specialized or tailored responses for different types of stresses. And then some threshold needs to be some or some critical points need to be passed. And then when plants kind of decide to prioritize one response over others, if this decision is made, then one kind of stress response may negatively influence the other. So this can happen. And importantly, also the functional links between like PTI and ETI is well established. But it seems, and probably everyone agree with me that so ETI can somehow exceed some kind of limit or threshold of PTI to further strongly activate immune response. And then once this happens, I expect that this may negatively influence the salt tolerance. So consistent with our data that so PTI triggering bacteria, so enhance salt tolerance, but not ETI triggering bacteria. But importantly, to my knowledge, we don't know the molecular basis for that kind of threshold, making, and also how plant immunity go beyond that threshold.
Yes, so many questions. I'm going to turn now to one in the Q&A box from Saumya Pusapati, who says, thank you for the very nice talk. I'm curious to know if you have tried inducing some kind of wound to observe the same effects as the increase in salt tolerance. I think that you see with the PAMPs. MAMPs.
Thanks for the question. The answer is no. We haven't really tried wounding effect on salt tolerance, partly because we didn't have a clear idea how to nicely set up wounding pretreatment, not to damage plants, but to really enhance stress tolerance. But the principle, yeah, I'm not surprised if you nicely show that effect.
Exactly, with it going through the whole PEP receptors going through SA and JA certainly makes it seem that maybe some wounding could induce that. So it's an interesting question. I am curious about the observation while we're waiting for some other Q&As to show up. I'm interested in your observation that about the sensitivity of the BAC1 BKK1 mutants to salt stress in the absence of added peptide, and what you think is going on there.
So yeah, part of the reason behind this we expect is that the environment of endogenous ligand receptors, which are dependent on BAC1-5. So please note that BAC1-5 is specifically impaired in so-called pattern receptor signaling function, not brachiostylide or other different functions of BAC1. So this implies the environment of pattern receptor like signaling machineries involved under salt stress, even you don't exogenously supply high dose of MAMPs or DAMPs. And this is partly shown in our study that indeed, so one such endogenous peptide receptor system, PEP receptor pathway contributes to basal salt tolerance.
Yes. Yeah, interesting. So Kenichi, I'm going to turn the next question over to you.
Right. So the question is pro-PEP1 overexpression, and pro-PEP1 overexpression increased root size and growth in normal conditions. So is it related to like a salt tolerance enhanced by this DAMP signaling?
That's interesting point. Yeah, actually, it's sort of described that no one may stress it out, and this may be also consistent with the observation made by general non-self-response paper. So in which the mutants seems to also grow poorly in kind of native soil settings. So yeah, I'm not sure whether the same thing happens between salt stress condition and such kind of native soil condition, but I'm not surprised, or I expect that. So these such endogenous, or even MAMP receptors involved in when plant growth on native soil in which many microbes are there. And then we also observe that BAC1-5 or BK plants grow poorly in Japanese soil. I'm not sure if this is true in all over the world, but yeah, we observe that too.
Yes. Okay, our next question is from Tianjiao Lu. Have you tested SAJA and ethylene defective mutants separately, but not their combined mutants?
Yes, we tested their mutants separately, and essentially they all retain PTSD. But we did it in a very kind of small scale in a kind of pilot study. So that's why we didn't include the data in the paper. But yeah, the single knockout mutants, yeah, like wild type in our settings.
You did try EIN too. I remember seeing that. And that seemed to have no effect. So,
right.
Yeah.
But of course, I mean, as I mentioned, in Wacky 1840 double mutants, in this setting, we need to be very careful about the seed quality. So if you compare like white type, fresh white type seeds and some mutant seeds quite old of your interest, then it may mislead you. So some really seedlings from poor, quality seeds tend to be sensitive to salt stress in this kind of acids using young seedlings. So please be careful.
Interesting. Do you think that has anything to do with abscisic acid? Certainly when we've seen variation in seed in Medicago, it seems to sometimes be caused by different growth conditions. So if we grow them in a more stressful greenhouse environment versus the babying them in the growth chambers, that we see different sensitivity to ABA. So I know that seed dormancy works differently for Arabidopsis. So I'm curious whether you think in those cases it is ABA, because you ruled out ABA synthesis with the ABA2 mutant. I wonder if you could speak to that possibility.
Well, first of all, so this PTSD seems to be quite robust. And then, so the loss of ABA alone, I mean, plants still okay. And then, but when it comes to kind of different dependence on ABA in a kind of context-dependent manner, so ABA response are highly also influenced by water status and also nutrient status. And then usually people don't care. I'm afraid to say so, but yeah. So then such kind of non-recognized conditional differences can influence your result.
Yes, definitely.
Okay, so the questions on Q&A. So PTSD might counteract the trade-off between defense and the growth and development in some conditions. So do you think this will kind of open up the ways to interfere with the negative effect of trade-off between plant defense and growth?
Well, yeah, actually, so we have a kind of a different study, I mean, independent, so not different from this work to pass the kind of alleviation of growth defense trade-off. But in this setting, please be careful. So we also show the seedling growth is better retained in pattern pre-treated seedlings compared to mock controls. But this is still, the growth is reduced, of course, compared to the mock plants under non-salt stress condition. So in this setting, so somehow growth inhibition, I don't know if I call this, but somehow the reduced growth is not really required, but something similar or a process associated with this may play a role in promoting salt stress tolerance. I don't know if I answer nicely to your question, but in this setting, yeah, if you compare between mock treated and PEP pre-treated plants under salt stress, yes, the latter PEP pre-treated seedlings retain growth better. But compared to really mock and non-stressed condition, so their growth is reduced. So I don't know if you see that alleviation, growth difference trade-off, but yeah, this is our observations.
So I have questions about the mechanism of PTSD. So you showed the salt stress, like a pattern triggered immunity, sort of a triggered similar response. And also in combination, there are some specific genes that are sort of become responsive.
That's
so
right.
do you think it is common genes are important for this PTSD, or it is a specific sort of a responsive genes in combination is responsible for this PTSD phenomenon? So what's your opinion?
Well, given the high robustness shown in PTSD in our setting, so I expect that so many kind of different components possibly collectively or redundantly contribute to PTSD. And then so somehow we have no clues, or almost no clues, so how plants acquire such high sensitivity or responsiveness, by involving otherwise non-inducible genes in salt stress responses. But yeah, I expect really some complicated or kind of many factors that collectively contribute to that. And then, as I mentioned earlier, so this is related to kind of priming. So plants indeed have ability to commonly induce kind of some core set of stress responsive genes or transcriptome, and then followed by more specialized, tailored responses to different types of stimuli. But somehow this kind of pre-stimulation of initial wave of stress responses allow plants to prime or establish some kind of primed condition. And then plants are better, can say more, okay to different stress, subsequently applied. So, so I have a feel, of course, this I would say just tempting to speculate that plants get ready for different types of stress or stimuli. It's so in this sense it's quite general. So that's why I expect that so many different components collectively contribute to that, and really depending on the context. I mean, the next stress, which next stress you expose plants, then what factors play a role would be different.
I love this idea of cross-priming of abiotic and biotic stresses. Really fascinating.
So nice word. Yes, cross priming. Yes, that's right.
Yes. Do you imagine any potential applications in the field at any point with these peptides?
Well, yes. Sorry, I didn't have time to mention that. But we have the observation similar. We see the similar conclusion also in rice as well. So this PTSD, in the end of this paper, we mentioned that so this is activated when plants face to possible alterations in plant inhabiting microbes. But this also predicts that PTSD should also involve such microbe or microbiome. So I have kind of speculation that this like PTSD can also involve such microbes, and then this may also provide a nice way to manipulate the microbes, and then altogether to enhance the stress tolerance in the crop plants as well. So this is the basic principle, and then. But of course, in the field, you cannot really supply lots of peptides to the corn field, or so instead, so you need to somehow stimulate the immune receptors possibly through nicely manipulating microbes or whatever, then you can also see the enhanced state of kind of stress tolerance to different types of stress. This is my prediction.
Yeah, fascinating.
Thank you.
Do you have any last questions, Kenichi?
No, I think I ask. Yeah.
So many interesting ideas and lots of good questions came up. I think, why don't we stop there then? And thank you, Yusuke, for a really fascinating talk. It's clear it's going to be very exciting to see the direction of this research going forward. So thank you so much for joining us with What's New in MPMI, telling about your study, your research, and sharing that with our audience.
Thank you very much.
Okay.
Yeah, great. Thank you. And thank you very much for the audience and really interesting questions.
Yes,
and
always good questions.
If you have more questions, please feel free to ask me. I don't know, directly or through the editorial office. I don't know, but yeah, welcome for further questions and inquiries.
Wonderful. And, you can find, I think Karen can type into the chat where you can find the link to the paper, but of course you can find it in our most recent focus issue. And of course, the authors always have their email address associated. And this is just a great example to show that the conversation doesn't stop here, but can keep continuing with the author as you have questions that come up from this.
Also, YouTube comments, right?
Yes. Also, and you'll be able to find this on the recording should go up and give us about a day, because as Yusuke and Kenichi and many of the audience are just starting their day, we are ending our day. So we don't think that we'll get that up until tomorrow, but there will be lots of opportunities there for people to watch and to comment on the recording as well. Okay, well, thank you so much. Appreciate everybody joining us, and for Yusuke for his seminar and Kenichi for joining me as co-host. Bye, everyone.